Hi everyone, welcome back to my channel and you're watching The Brown Feminist. In today's video, I'm excited to bring you another crash course. And this time, our crash course is going to talk about how exactly to conduct a clinical trial that is investigator-initiated or physician-initiated. Now, first of all, let's talk about what that means and what this crash course is going to help you with. Firstly, a physician or an investigator-initiated trial is one that is run exclusively at the site level, meaning it is run at a hospital, clinic, or academic setting or a research institute. And most typically, the kind we are going to focus on this one is ones that are actual trials, which means they're usually interventional. There's some kind of drug or surgical or procedural intervention or observation, and they're typically conducted by investigators who are physicians or clinicians. So these clinician investigators are most commonly, especially in the North American markets, they're usually doctors, so they're usually medical physicians with an MD. However, in more recent times, we do see a rise in clinician investigators who are NPs, nurse practitioners, clinical epidemiologists, and others. So in this video, what I'm going to help you is to get a full and thorough understanding and an overview of everything that works and how it all happens when you are working as a research coordinator or a research associate or a research assistant in the scope of an investigator-initiated clinical trial. Previously, you must have seen my general crash course, which had a goal to help you to do interview prep. And in that video, I mostly focused on regulatory pharma-sponsored trials. If you do want to do a crash course on that, and I have heard so many comments from all of you guys about how that particular video helped you guys land your first dream role and ace your interviews and handle all of the questions coming from those interviews when it came to roles that focus on pharma-sponsored regulated trials. Now, a lot of you since then have made comments on my videos and sent me emails and messaged me on Instagram and other social media platforms and asked me to make a similar video, but this time focusing on investigator-initiated trials. So here you go. This is your full and final crash course that's going to cover every little element and everything that you possibly need to know in order to enter the industry, in order to ace your first role as a research assistant or a research coordinator, even a research associate at the site level under investigator-initiated studies. Now, as I do this video, I will also be making little comparisons about what is different for each of these steps between pharma-sponsored studies and investigator-initiated studies. Ideally, when you are entering the job market, you would have done both crash courses so you can handle all kinds of questions in your interviews. It's also really good to have skills that span the whole spectrum of both of these kinds of clinical trials. And of course, there is another whole niche of clinical studies which are not trials, but they are more observational studies in clinical research which happen more in academic settings. Now for today, in this crash course, we are going to focus on the trials, typically randomized controlled trials, placebo-controlled trials, double-blinded or single-blinded studies or unblinded studies within more of a clinical area, within more of a hospital or clinic site level. So if those are the kind of jobs you're applying to and you're new to this field, or you just want to gain some extra clarity and knowledge about how investigator-initiated trial studies are run, then this is the crash course for you. So I hope you enjoyed it, and before you continue, don't forget to hit like on this video and subscribe to my channel. By subscribing, you will be supporting creators like me who have a day job and career in clinical research, but also like to have a side project of sharing my knowledge with all of you guys and helping to build a community where we can inspire aspiring clinical research professionals such as yourself. So these subscriptions do give me added motivation to make more videos like this. So once again, please take a second to subscribe to my channel if you have not already. You can also follow me on all of my social media platforms that are listed below. So without further ado, let's get into it. In the first part of this crash course, we are going to talk about protocol and plan development. Now, if you are familiar with pharma-sponsored studies that are regulated, then this section is actually very unique to only investigator-initiated trials. And the reason behind that is that in case of pharma-sponsored studies, those of us working at the site level or the hospital or clinic or academic level receive the already built and designed protocol, plan, and documents from the sponsoring pharma company or from the supporting CRO organization. So this is not a part that we need to do. However, the very basis of an investigator-initiated or physician-initiated study is that the actual clinical team comprising of this physician, this doctor, as well as the research team actually come up with the design of the study and every little logistics setting of how it's going to be conducted, the logistical needs, the plan for collection, the team of scientists that are going to be part of it, selecting the sites, and all of this stuff. So it all basically begins with one core idea about the research hypothesis. Now, more often than not, it is usually exclusively the physician and maybe some co-investigators, maybe an associate that works directly in order to design the research protocol. This protocol requires a lot of knowledge in the field that is usually more than logistical and coordination knowledge. So typically associates have a little bit more in-depth knowledge of the field, all the literature, what more needs to be investigated, what needs to be compared, the kind of controls that need to be in place, how best the study design will work. At this phase, they also sometimes involve people like statisticians, analysts, and stuff in order to come up with the ideal sample size and see how many samples or what types of a patient population they actually need in order to prove or disprove the hypothesis, what exactly they are testing, and all of these designing elements. This is what makes up the core of a physician or an investigator-initiated trial. Now, while they're doing protocol design, they usually come up with a very short and simple document which outlines how they intend to do it, where they intend to recruit from, how they will ensure that the target population can be acquired and recruited from within that geographical setting or that hospital site, or the fact that they plan to involve other sites in recruiting these patients, and so on. At this time, the investigator also chooses to usually form professional affiliations with other PIs who can become co-investigators of the study. These other people can be academics, they can be other physicians, they can be physiotherapists, nurse, other forms of clinicians, and maybe people who have certain expertise in the subject matter or people who have access to the patient population in another clinical setting. So once all that is done, it is usually only the research associate and the investigator who develop the original document. However, at times the coordinators are also consulted, the research assistants are also consulted in terms of the logistical needs of the study. They can provide input about, okay, is this actually going to need certain unique ethical approvals or certain additional elements to go through ethics smoothly? Is it going to be realistically possible to implement at the site level? Will certain elements of the study have to be outsourced to third-party vendors? What kind of shipping supplies are needed, and so on? Once that is done, this protocol document will also contain a study budget that is proposed after discussion with the immediate research team. And that usually concludes the first part of designing and working on a clinical or an investigator-initiated study or a physician-initiated clinical trial. It all starts with this one document, which can be anywhere between 2 pages and 5 or 10 pages, which has the outline of the protocol, the collection of data, why the study is being done, what kind of population or sample size is required, what kind of budget they're looking at, and what can be the ultimate gains or benefits coming out of this study, which makes this so vital to conduct, and what are the potential risks and harms. So all of this is actually summarized in this one protocol document. So going forward, that protocol document is shared with members of the team, and it becomes vital for everyone to refer back to it to see what the original study design was. It also contains information about the study timeline. If it's a study lasting a few weeks, a year, 2 years, 5 years, 10 years, and of course that in turn can, require or have an impact on the kind of budget the city needs, the kind of human resources required, clinical staff requirements, the number of patient visits, and so on. So that was it for our part 1 of Investigator Initiated Clinical Trials. Now, without further ado, let's get on to part 2. Now, part 2 of any kind of investigator-initiated trial requires grant funding. Now, unlike pharma-sponsored companies where the funding usually comes from a private entity such as a pharmaceutical company, those usually have an abundance of budget as well, with lots of negotiations going on between the site and the pharma. But compared to that, these kind of studies have more tighter, more restricted grant funding, which usually comes from, a taxpayer-funded grant, a government-funded grant. There can be some smaller nonprofit or private-funded grants. So what kind of grant is going to be applied to are all decisions that the investigator themselves and maybe some co-investigators come together and decide for that kind of study. As a research associate, this is an important part for you to be familiar with. Clinical research associates at the site level in particular begin to have some kind of supportive scientist role, which means a lot of the times you can be heavily involved in things like grant writing. Most typically, a clinical research coordinator or a research assistant does not usually have a big role to play in writing the grants. However, supportive work might be needed. It really depends on the options and on the design of the team, on the relationship that you have with the PI, on your eagerness to support the grant. Sometimes even as a research assistant or as a research coordinator, I did raise my hand and volunteer to be a little bit more involved in grant submissions and to help select grants and to help assist in the process. All of that was really excellent for me to keep learning how this process works. And any extra knowledge, of course, is never a waste. So for those of you who do want to work as an associate at the site level, this might be something that you want to start volunteering to help with. On the other hand, if you are planning to purely work as a research coordinator in the long run and stick to the administrative side of the work, or eventually in the long run you want to transition to a CRO or pharma, then learning how to do grant submissions at this stage may not be the most helpful to you, although I will still stick to my typical saying and say that no additional knowledge in your field of work is ever wasted. So when the investigator and sub-investigators and research associates and co-scientists decide where they're applying for a grant, they have a wide variety to choose from. Sometimes even pharmaceutical companies give out grants. They can be restricted or unrestricted finances. Sometimes these can be non-profit organizations, development organizations from across the world. There can be charitable foundations who are funding these research studies. A lot of the time, private entities fund them as well, but it is also far, far more common, especially in Canada and the US, to have those funded by the government. In Canada, one of the main federal funding agencies is a CIHR. If you haven't heard about them, I encourage you to look into them, just Google them and learn a little bit about them to have some general knowledge about who they are and what they do. But they are our main funding research, research grant funding organization. In the US, it could be the NIH, the National Institute of Health, and so on. The FDA could, fund certain internal research, but also hospitals can fund research, and different kind of research networks can fund research. Sometimes the government will give a lump sum of money, like $5 million or $1 billion or whatever, and allocate it for, say, hepatitis C or HIV or cancer, and give it to like a sub-organization that works as a non-profit, and they might then subdivide it up and run little grant competitions and then reallocate that to fund both observational research, interventional research, clinician research. Sometimes there are departmental funds and grants available to people conducting research within certain hospital specialties. So there's really a lot of different kinds of grants to choose from. But this step is especially important because in 95 to 99% of cases, but don't quote me on this number, but in majority of the cases, if your research is not getting funded, then it is not moving forward to execution. A lot of studies wait months or years before they can be properly funded. On the other hand, sometimes PIs might have some discretionary funds available. They might be able to pull funds from a common pool from other research projects and re-divert them, so at least the projects can get off the ground and do a pilot phase of the study or do some preliminary work while you're waiting for more abundant grant funding to come through to support the full scope of the trial. So that basically concludes our part 2, which is about grant funding and the budget, which really determines the capacity to complete the research in a timely manner and to full completion, be able to actually recruit all the patients, hire all the staff needed, involve all of the sites, and buy the materials that may be needed, pay for the testing that may be needed, and all of these things really depend on the grant funding. And that ends part 2. So without further ado, let's get to part 3. In part 3 of a typical investigator-initiated trial, we see that you begin recruiting at the local site, but you also do things like site selection and activation across multiple sites. Now, this depends on if your study is a single site study or a multi-site study. Typically for larger, sample sizes, maybe you have to recruit 500 patients. So if you're doing it only from one hospital in one city, it might take a much longer time to recruit that level, that number of people who meet all of your inclusion-exclusion criteria. But people don't want to take that much time. So if they can afford it, investigators will reach out to their peers in the community who maybe work on the same specialty, have other sites, could be other sites in the same city, same province, state, country, even internationally. They might begin to partner with others and liaise with others in order to have additional sites contributing patients into their trials. And by doing so, there's 2 benefits that happen. One is this research doesn't take years to happen, which means you're not waiting years and years to find out if your hypothesis is proven and disproven. There's also a risk in that scenario. If you're waiting 5 or 10 years to conduct trial, not only are people being deprived of some potential good outcome of your research, but you're also risking your career because other people might find this out. People with better finding might have similar ideas of research and find out the results and publish it before you, in which case the value of your design, the value of your research is much less. But then on the third situation, that's also just more beneficial because it's more efficient, right? You're not spending so much time waiting to recruit from one place. You're also building collaborations, and you're allowing patients from a broader geography, from multiple kinds of sites, to benefit from this potential research. So that usually is something that happens in part 3 on this phase, that you start building these collaborations. But this is also the phase where clinical research coordinators or project managers begin to get more involved in the study. Now, at this point, the scientific aspect is pretty firm. You have written a grant, you have written the protocol, you have a plan of the full methodology, you have done your sample size calculations, you know exactly what you want to do. Now, you have to then go ahead and decide how it's going to be done, which sites are going to be involved. In order to do this while physicians themselves do play an important role in, retouching up their network and just, sending out emails and messages and saying, hi, this is the study I'm doing. Would your site like to participate? Their networks and relationships play a big role here, because those other sites may just be contributing patients and may not gain, scientific publishing from here or other say. But usually physicians and investigators and clinician investigators in general have a peer relationship that they will do this for each other in order to grow in the research community. So as they're doing this, the coordinators will then start to be tapped in and say, let's begin activating the sites. So site activation is a very, very important part of this step as well. When your sites are selected, especially at your home site, let's say this is not even a multi-site study and it is only happening in one site, which is your site, the lead site. Even in that case, site activation means getting every single thing ready in order to actually now run this research. So you have your protocol, you have received your grant funding, but next you need your REB approval. So whether it's at your site or at other sites, you need to make sure everything is in place in order to get your REB approval, which means documents such as the protocol, documents such as the budget, documents such as the informed consent form, materials that will be used to recruit patients, email templates, any kind of data capturing plan of how you want to capture your data, in what format, what kind of exact data will be captured, what kind of personal health information you will need access to, how many trained personnel will be there, who will be conducting the clinical portions of it, will there be a hired nurse, or is the researcher themselves going to do the procedure, is pharmacy department going to be involved, is the diagnostics division of the hospital going to be involved, are third-party vendors going to provide some services here. So all of that needs to be determined at this point and then informed to the REB through an initial ethics submission. Now, at this point, unlike part 1 and part 2, at this point, we are again becoming similar to a typical pharma-sponsored study. In a pharma-sponsored study, part 1 and part 2 is done for us by somebody at the pharmaceutical company who have they have their budget, they don't need a grant submission, they have their protocol and all of their documents ready, they have done their site selection, they have done their negotiations and budgets, and now we're talking about site activation. So part 3A was site selection, but when you don't need site selection, you can move directly to part 3B, which is site activation. Now, site activation is chief and primarily the job of the research coordinator or the study coordinator. It can also be the job of whoever is the clinical research assistant. Sometimes it is the job of a clinical project manager. Some studies which are larger, like multi-center studies, they might have one central manager who helps regulate things across all the sites, but then each site might still have their own study coordinator. So this is a very important role. This is the point where if you want to know a little bit more elaborately, I will be linking down my other video which talks about pharma-sponsored studies, because this step is very, very similar whether you're doing investigator-initiated or pharma-sponsored, because the site has to be ready, and that means paperwork needs to be ready, a plan in place needs to be ready, things need to be safe, research personnel and clinical personnel need to be trained, and all of their training, degrees, licenses, and certification and resume need to be filed, right? And there needs to be enough ways of reporting if anything goes wrong, whether they are SAEs or AEs, adverse event documents, protocol deviations. There has to be a data captured plan. So this is all I'm going to say for part 3, but in part 4, we are going to go a little bit more into what data capture planning and actual data capture really mean in the case of an investigator-initiated study and how it differs from a pharma-sponsored study. Without further ado, let's get to part 4. For part 4 of this crash course, we are going to be talking about the DCP and the DSA. So a DCP is essentially a data capture plan. Once again, this is very different for a investigator-initiated trial versus a pharma-sponsored trial, because the pharmaceutical company had their internal scientists and clinicians already tell you exactly what kind of data needs to be captured. And they usually have built their own kind of electronic database, which may be called an EDC, an electronic data capture tool. Sometimes they look like little forms electronically you have to fill out, or they can be surveys that patients themselves have to fill out. It can look like a variety of things. Sometimes they can be actual tests and blood work and other kind of data variables that need to be captured. They can be in video format or image format. They can be MRI scans, or they can be things you actually qualitatively ask the patients and input into the system. However, in the case of an investigator-initiated study, all that is determined by the study team. At this point, the study team comprises of both the investigator, the clinical research associate, and to some degree, the clinical research coordinator. Primarily, the physician, the co-investigators, and the clinical research associate are going to get together and determine exactly what wording to use, what format of data capture to use, how best to capture the data that they are trying to get. What kind of testing will need to be done, what kind of baseline and follow-up tests they will need to do, what kind of questions they need to ask the patient in order to fully capture all the variables they need to prove or disprove the hypothesis in question. They will also decide on things such as drug dosage and other little details and ways to capture any kind of variability and ways to account for control groups and other things. So at this point, you might have to assist in things called DCPs, which are data set creation plans. This is typically not created by a study coordinator, although you should have good knowledge of it. You should know exactly what information ultimately needs to be captured. However, the design of this is usually done by any kind of associates, co-scientists, assistant scientists, physicians themselves, co-physicians and investigators supporting the study, could be fellows and residents, specialists involved. Everybody can play a role in this. It really depends on what the research team looks like, who's conducting the study. Once it is done, however, it is definitely shared with the coordinator and the rest of the study team. Sometimes it can be something like a simple Excel sheet. Sometimes it can be a digital survey, but sometimes it can require an eCRF to be built, which means an electronic case report form might need to be designed. At times, the coordinator may have input on the usability of it, how user-friendly it is, and you can provide input on, data safety and security, and say, patients will find this difficult. Because a research coordinator or a research assistant have a lot more knowledge and experience with the front side of this. They're the ones that patients will come to and complain, or come to and share their challenges and difficulties. So when it is being built, sometimes it is good for coordinators to have that relationship and go back and forth and say, I know you've done this, but our patient population is all going to be 65 plus. Many of them will have some limitations in accessing technology. So unless we are giving it to them on an iPad when they come here, or we are guiding them through it, it might be difficult for them to fill out certain forms. Or you might actually bring in some interesting ideas and information. Like you can say, what about people who have hearing disabilities? What about people who have visual disabilities? What about people who have language barriers? How are we accounting for them? At the end of the day, the study coordinators are responsible to meet these high ethical standards, and they have the most knowledge of the front end, of the patient side roles. So it's really, really good and better quality design of DCPs when the coordinators and front-facing roles, whether it's a nurse, it's a coordinator, it's a research assistant, but when these individuals in the team are involved in the DCP. So when the DCP is done, next we have to talk about data sharing. So not always will you need to have a DSA or a data sharing agreement. A data sharing agreement is only required, and it is basically a legal contract that is only required when important data that might be healthcare data, can be personal health information, can be anonymized health information, but any kind of health information is being transferred or shared between institutions that are not both covered by the same REB. So in the case that you are actually sharing data between sites, between cities, maybe your study is a multi-center study and all of these 10 sites are going to send the lead site all of their data, whether they're sending it digitally through the ECRF, or they're sending it in an encrypted Excel file, or they're sending it in any other way through a secure link, a secure server link. All of this still has to be done only after a DSA has been signed by both parties and it is active. So usually you will have some help from the legal department, whoever your legal consultant is, your counsel is, to help draft the template and modify anything within the DSA, the data sharing agreement. However, it is important as a physician or as a associate or study coordinator or research assistant to have knowledge that these things need to be in place in order for ethics to agree that this is not an ethical violation, in order for ethics to be sound and maintained, in order for you to share any kind of data outside the organization. Unless this is in place, you just sending somebody information about your patients, even if it doesn't have direct identifiers, it is very, very unethical and can lead to malpractice and many other legal issues. So this part was all about part 4, where we talked about the data capture plan, the data capture method, as well as the data sharing agreement when data is shared between different organizations. One last example about the DSA, the data sharing agreement, is about how when you are actually sharing the data, it doesn't necessarily have to be from other sites to your sites. Sometimes you might even share data between different PIs. Sometimes maybe all the patients were recruited at one site, but you have outsourced a part of your analysis to another organization. Even in that case, as long as data is moving between institutions, you have to have a data sharing agreement in place. So I hope that part has been clear. Now let's move on to part 5. In part 5 of our crash course, we are going to be talking about recruitment, enrollment, informed consent, intervention, and visits. Now again, this part is actually very, very similar to the ones that we do for pharma-sponsored studies. So if you want a more elaborate video on this and know all the nitty-gritty things, please go and watch those sections in the crash course where I talk about a typical clinical trial, clinical research crash course for pharma-sponsored studies. I am linking it up above here in the video as well as down below in the description. I hope you will find that helpful. Now here, just as an overview of what happens here, it's very similar no matter what kind of study you're doing, whether it's investigator initiated or pharma sponsored. But essentially, the study coordinator and research assistant are directly responsible to recruit patients. Usually you have already shared your recruitment plan. Are you planning to approach people who are coming to clinic? Are you planning to approach people who you have screened for eligibility from a central hospital database? Whatever that might be, you're planning to reach out with them through email. Whatever method you might have for recruitment by now will have been pre-planned and approved by your REB and your team. So you will simply follow through with that plan and actually perform recruitment where you reach out to patients, you're sharing what the study is about, telling them about the risks and benefits, and if they do consent to participate in the study, you're performing the informed consent process. Once the informed consent process is done, you are enrolling them into the study and then doing the study procedures. If this is a randomized control study, you might perform the randomization part, or the randomization might be done by somebody at the lead site or somebody who is a central project manager. Either way, once randomization is done and you know what intervention they're going to get, the study coordinator has to be very cognizant about the study methods and protocol. For example, if it is a double-blinded study, you must be very careful not to reveal the randomization to the patient or to the investigator or anyone that's performing the interventions. For example, I was part of a study where it was a placebo-controlled randomized controlled studies. And for the full 8 weeks of that study, I could not tell neither the physician who is performing the intervention and giving the drug, nor the patient what exactly they were getting. So at my site level, I was the only person who knew if they were getting a placebo or getting the drug. And I was also responsible to collect it from pharmacy, to bring it, to do any kind of processing, to prepare it, to ensure all the labels are intact so you cannot tell them apart, and then present it to whoever, like the physician who is going to administer this drug. So only after the time period has elapsed, and if the study protocol permits, can you actually unblind people. So this is just one of those examples of how the methodology and all of it needs to be really crystal clear in the mind of the study coordinator or the clinical research assistant. Your job is also to make sure the interventions and follow-up visits are conducted in time. All the data is being captured in a timely manner and in an accurate fashion. All the documentation is on point, and of course, advocate for the patient. If they have concern, they should be able to reach out to you. Any kind of problems that may arise, you will have to learn to troubleshoot. I hope if you want a little bit more elaboration on these, you will consider watching the other crash course, which goes into much more detail about the informed consent, patient visits, and all of these things. So that basically concludes part 5 of an investigator-initiated clinical trial. In part 6 of this crash course about investigator-initiated clinical trials, we are going to talk about what happens once data quality is in question. So of course, this is something you can absolutely implement from the very first time. For me, it was a learning experience as I did more and more clinical trials, and there were more mess-ups. So typically, for a well-designed clinical trial, you will have certain data quality assurance and data quality monitoring steps in place. What that means is, in order to ensure that RAs, research assistants, or coordinators across all sites are collecting the data in the same way and there is least amount of variability, you can do certain things such as site training. If you are the coordinator or the associate at the lead site, you can actually propose a central training session where all the site RAs, all the sites coordinators, anyone who's involved in the study comes and they get an overview of the study. They get to ask all of their questions. They're given all the best practice rules for these, what to do, what is the ideal way to go about doing it, what is the ideal way of recording data. This is something that can actually be done throughout the life cycle of the study. So not only should it be done once or twice at the beginning to ensure all of your staff across sites and within your site are trained well and prepared to embark on this study, but you can touch base every quarter, every 6 months or so, every year, whatever works for your study, in order to see if there has been turnover, if there is new staff who need training, or if there are common problems that are arising and common questions that are coming back. By clarifying their questions, you are actually ensuring that your data quality is not going to be hampered. But there are also other ways to actually do checks. So you can do something called central monitoring. This part in particular is significantly different between a pharma-sponsored study and an investigator-initiated study. In pharma-sponsored studies, the training is usually always provided by some member of the pharmaceuticals company to the site. In an investigator-initiated study, it's typically done by the lead site, which is a site who came up with the study, who received the grant funding for the study. It can be anyone at this site. If it's something more clinical, then it's more likely that the investigator themselves might want to do the training. If it's something a little bit more logistical, more in-depth, it can be the clinical research associate or the clinical study coordinator who actually takes these training sites. If it's a big national study, then and it has a central project manager, then they can do the training. But there is also an element of study monitoring. Now, if you don't know what monitoring is, once again, it's a bit different between regulated pharma-sponsored studies and unregulated physician-investigated studies. But essentially, monitoring is a kind of auditing where you're going to ensure quality and make sure quality is high and there are no irregularities, the data collection is proper, it's accurate, and so on. In order to do this, in case of pharma-sponsored studies, it's usually a study monitor, or a clinical research associate who works for the pharma company or the CRO, who goes from site to site to perform these monitoring visits. And they're very serious. This is regulated. They have rights to shut down the study if it is not being done properly. And the risks are also higher because sometimes they're more phase 1, phase 2, phase 3 experimental drugs and so on. In case of investigator-initiated studies, the risks are sometimes lower. Investigators typically do a lot more stage 4 studies, which means they might be doing clinical trials with slightly modified existing research methods or slightly different combination of drugs that are otherwise already approved, trying to find the best doses, trying to find the best combination of medications. But usually these medications are not fully experimental and they're already approved for use in that population. Or maybe they're approved for use in the general population and they're testing it in a specific population. So while the risks are lower, study monitoring is typically still done, although it might be virtual, it might not always be in person. Now remember, these studies are not funded as well as pharma-sponsored studies, so it is not always possible to have an associate at the read site who can travel by air or travel by road and have a hotel and go to places to do on-site monitoring. But a lot of the time it is done centrally, virtually. They might ask to see certain documents. They might walk you through certain processes. So this is how this portion is significantly different between pharma and investigator-initiated studies. But essentially, step 6 is all about quality control and quality assurance of data collection. Now, data collection, a big chunk of work for the central project manager in multi-site studies is also to ensure sites are getting activated and recruitment targets are being met. When recruitment targets are being met, that visits are being done in time, things are moving along, everyone has the training that they need, all the problems across sites are being solved. For you, as somebody who is a study coordinator or research assistant, this is the heavy part of your work. This is the part where you are actually going to be doing all of your recruitment, follow-up visits, blood draws, whatever else needs to be done for that particular study protocol. And soon enough, your time will run out. You will have to do REB renewals, continuing reviews, submit any kind of protocol deviations or adverse event reports. You have to make sure all of the ethics stuff is up to date and maintained. That brings us to the end of part 6. Now, part 7 is post-study. This part is also significantly different between a pharma-sponsored regulated study versus an investigator-initiated clinical trial. The way it's different is that typically once all of your patients have been recruited and drugs have been given, follow-up visits are done, data collection's done, then in a pharma-sponsored study, your job as the site is done. You submit everything over to the pharma company or to the CRO. They do a final monitoring visit, which is the study closeout visit. You make sure all invoices are paid and your work is done. However, for investigator-initiated studies, the work is still just getting started. Once you have collected all of the data that you need, it goes back to being the research associate, research coordinator, and the PI's job to now work on the data. A lot of the time, they need to work further to make sure data is complete and then perform data cleaning. Some degree of data cleaning can be done by the study coordinators, and data cleaning can mean anything like making sure they're all in the same format, they're all making sense, nothing is incomplete, they're all in numericals, they're all downloaded in one place, formatting and all of that. But a lot of it can also be done by the associate. So the clinical research associate and the PI may be working on the data to make sure it is now ready for analysis. Sometimes the analysis is done by another person in the team, such as an analyst, a biostatistician, and so on, depending on how complex the data is. On the other hand, there might be simpler analysis that can be done by the research associate themselves, especially depending on how much background they have in statistics and epidemiology and different kind of analytical tools and softwares like R, programming languages, SAS, SPSS, and so on. Sometimes the PIs themselves prefer to do parts of the analysis or be directly involved in this process. So at this time, you are starting to keep in mind that you are trying to find out the results and prepare some kind of a manuscript to publish and share what has been done in your study. So this takes weeks to months. Sometimes it can take up to 6 months or a year, depending on how many competing projects there are that also need your attention, but also how urgently you want to push this out for publishing. And that concludes part 7 of our crash course. Now, for part 8 of our crash course, we are going to talk about the closing up of things by the study coordinator. At this point, the study coordinator should have all the documents in place. Now, one thing people get confused a lot is whether or not you still need ethics if you are not actively recruiting patients. In theory and in my knowledge, you still need to have an active research ethics application open and continuing reviews done as long as you are still working with that kind of study data. Until the data has been published and the study is fully closed, you really need to be involved. But there are also other documentation to keep in mind. For example, there can be things like, investigator brochures that you've sent out or DSMB reports. A DSMB is a Data Safety Monitoring Board. Some trials, both pharma-sponsored and investigator-initiated trials, may form a Data Safety Monitor Board who take periodic data updates from the central coordinator or the lead site, and they make sure that nothing unsafe is happening. And they don't need to be blinded. They get to see the true and raw research data as it's coming through. They might meet once in 3 months or once in 6 months on such a periodic basis and review the data to make sure the study is okay to still continue. And once they have met and had a discussion internally, they make a recommendation to the study team if the study can continue recruiting, or they need to change something in the methods, or the study has become so unsafe for the patient population that it needs to be shut down. So all of these documents also need to be submitted and maintained. But on top of that, the study coordinator's job is also to submit a study closure document and form to the local REB. You can also coordinate that with all of your other sites if it's a multi-site study, and make sure all your RAs have a debrief. You thank everybody and say what a wonderful job they've done. You can share some final numbers on how recruitment went across sites and in total. You can share some procedural data like that throughout the course of the study and especially at the end. But most importantly, don't forget to archive your documents. Whether it is all electronic data or it is actually paper data, you have to make sure that all of the data involved in the study at each site is actually saved and archived in a trial master file, TMF. So the TMF then needs to be put away for a certain number of periods during which time they can be audited, and they must be retained for that number of years after study closure for legal purposes. Sometimes that can be 10 years, 15 years, 20 years, 30 years. It really depends on your jurisdiction. So it's important that you speak to your local institution and find out for how many years you should store all of your research documentation, electronic or paper, and how it's going to be eventually destroyed. Make sure there is a plan in place. And that concludes our part 8. Now we're at part 9 of this crash course about investigator-initiated clinical trials. In this part, we're going to talk about wrapping up your budget. Of course, initially I had told you guys about how you're going to get your grant funding or some kind of budget source in order to fund your study. Now, by the end of the study, it is again the study coordinator's job or the research assistant's job to ensure all vendors and all sites have been paid. Oftentimes, when you're working in a multi-site study where all of these different sites across the city, across the state, or across the country, or across multiple countries are contributing patients and recruiting them for your study, they usually get a certain amount of reimbursement to cover the hours of staffing and other materials that they had to spend in order to help you conduct this study. Whatever of that amount might be and whatever the process of reimbursing them might be would have been disclosed previously when you were signing up your sharing agreements and contracts between your different sites. So you can refer back to them, ask for those invoices, and make sure all of them has been paid off. You also have to make sure financially the account is in good health to pay off all of your vendors. Now, vendors might be internal vendors, such as within the hospital. You might have to pay off the pharmacy department, or you have to pay the fees for the diagnostic division for spending all of this money on, conducting the tests for your clinical trials. In countries like Canada, which have universal health care, unless the tests done are for standard of care, If the tests are specifically done for research purposes, then they are not reimbursed by taxpayer funds or by the province, which means in those cases, it's very important to keep track of these and, declare when it is specific research related testing that you're doing and pay off that from the research budget. In the end, you might also need to do communication with your financial analyst or whoever your accounts person is and make sure, if the fund within the grant account is overdrawn or underdrawn, there's leftover funds. If any other way you can utilize that fund to finish up this research project, you have to ask for an extension for that fund. Funds themselves can have lots of different rules. For example, many grants will not allow you to take remaining funds and just invest in buying research equipment, right? They have to be pre-declared in the budget. So you should be using your research fund in clinical research to do exactly how you had set out describing it in the budget. And whatever may be left may be going back to the original grantor. So that concludes our step 9 and part 9 of this crash course. For part 10 of this crash course, we're going to go back to our PI, the original physician who had started the study, and any kind of co-PIs involved in the study, as well as the clinical research associate. So these 3 classes of people in the study team are now going to be working with the analyzed data. They're going to be writing up a manuscript, trying to submit it to different journals for publication, starting with slightly higher impact ones, seeing if they're appropriate fit. If they get rejected, you might go to a slightly lower impact one and continue until it is accepted and published. Sometimes there are also obligations set out in the original protocol that you will let your scientific community or your general public or your patient population who participated in your study know about the outcomes. If such a thing was in the original plan, at this time you might ask your research coordinator to reach out to all of the patients and let them know by phone or by email or by sharing with them a little newsletter summarizing the publication or summarizing the findings of this study. You may also wish from the study team to present this at different conferences, to share this with your granting agency, or share it with all of the co-authors and your peers in the community. And that brings to an end this crash course on physician investigator or investigator initiated studies. I hope this video has been helpful to all of you, and if it was, please don't forget to give it a thumbs up and leave me a comment below if you have any comments, any feedback, or any further questions. I would be happy to answer them. Well, this was it for today. Until next time, this is the Brown Feminist. Bye. Hey guys, before you go, don't forget that I do offer one-on-one career counseling services in clinical research. So if you do feel that beyond the videos and the emails where I answer all of your questions, you need a little bit more of hands-on support, you're not getting those interviews and you're not really sure why, or you just don't know how to handle certain questions because of your, you don't have all the experiences, but you know that you can do the job. If you need some additional help in resume building, cover letter design, interview prep, or just general career consultations, I do offer this service now live. Find out more by clicking the link below. I have a whole video where I explain what services I offer, and there's a form there if you would like to sign up. Until next time, this was the Brown Feminist. Bye!